Okay, hello everyone. My name is Matthew Kempton. So we're going to now be doing the interactive part. We start with a few slides. So there's a group of us doing this section. So I'm Matthew Kempton. We also have Mariana, Ruba, and Jack. So we're just gonna, if we just, oops, right, this one here. Okay, in terms of working with structural images, we have a number of metrics we can work with. We can work with surface measures. That's often through something like free surfer, volume measures, which may be the traditional ROI approach, such as some of the manual approaches, also VBM, free surfer also outputs volumes, subcortical and cortical measures. And we might want to look at the structural differences by the different participant groups. There's also another technique is voxel-based morphometry, which I'll touch on shortly, actually, in some of the analysis we are doing. And this was something that Simon touched on earlier, and Vina, I think, as well, looking at some of the kind of key findings that have been found in schizophrenia. So by far the largest studies really have been the Enigma consortiums. So the Enigma consortium looked at patients with schizophrenia, and they published two studies. So the first one was in 2016, where they looked at subcortical volume, and that had about 2,000 patients, 2,500 healthy controls. And on that graph there, you can see on the x-axis, we have Cohen's d effect size. So the negative effect sizes are where it's a smaller structure in patients, and the positive is where it's larger. So we can see the lateral ventricles were enlarged, and that was something that was found way back in that study that Vina quoted by Tim Crow and some of the colleagues. But obviously we've got a lot more data now, and we can see other regions are affected as well. So the way that these kind of Enigma papers tend to come out is it's a little bit based around the free surfer model and the output. So there's a subcortical volume stream and then a surface stream. So we've got the subcortical paper came out first, and by the time the second paper was in development, people have got quite interested in this. So we have more. That's why there's a kind of boost in the sample size. So we have about 4,500 patients, 5,000 healthy controls. So a sort of striking finding, I guess, is that it's very much affecting a lot of the brain rather than one key area. So there's, it looks like the whole of the cortex is affected. It's a little bit hard to tell from that color bar there, but the maximum effect size was seen in the fusiform gyrus with an effect size about 0.5. So those are the Enigma studies in schizophrenia. More recently, Enigma has been looking at the clinical high-risk state. So this is grey matter abnormalities. This is basically the same type of findings in the sense that they looked at subcortical volume and cortical thickness. So what we've got on the first row is clinical high risk versus healthy controls. So you can see the sample size is a little bit smaller, and this is generally because there is less studies or less samples out there. They're more difficult to recruit. And we also have the subcortical regions on the left, the cortical thickness on the right. So you can see, first of all, that the cortical thickness and subcortical findings are a lot less in terms of their effect size. So just looking at that color bar, it only, well, it probably goes up to about -0.2, but most of the effect sizes are around about -0.1. So really quite subtle effect sizes. And that's in clinical high risk versus healthy control. If we then move on to the second row, we have transition versus non-transition. Quite a relatively small number really of transitions, and this is partly due to the idea that around about 20% transition to psychosis. But you can see there it's really quite subtle. So I should say actually the grey areas are areas where there's no significant difference. So there's a number of regions that are non-significant, and the regions that are significant are really quite small. So we're talking about effect sizes about -0.05 or so. So really pretty subtle. Just going on, sort of a bit of a cheeky plug to some of the work that we've been doing. So we have been also looking at the Enigma sample, and these are people with early onset psychosis. And so we have, sorry, the formatting's gone slightly there, but there's 470 patients versus 480 controls. So sample size a little bit smaller, but these patients are very difficult to recruit. These are, well, they're quite rare. They're people who get psychosis before or the age of 18, many of them sort of 13 and 15 and so on. So this is using a different technique, voxel-based morphometry, using our Enigma VBM tool. And here we've got the color bar is saying Hedges G. So that is really similar to Cohen's D. So it's basically the same. It's just got a small adjustment for small sample sizes. And you can see, similar to the other work, that there's basically a lot of the brain is affected. And we saw the largest effect size. Again, sorry about the formatting, it's 0.54. We also looked at the effects of medication, chlorampromazine equivalents, and in these patients, we saw a reduction, particularly in the orbitofrontal cortex. I should say all these findings are corrected for multiple comparisons, so they're quite robust. And that was published molecular psychiatry by a PhD student, Shijing Si, for her PhD. Okay, just moving on to really the need for these large sample sizes. So this is a real importance of something like Psy-ShareD, where we're trying to pull data. So you can see from this graph that as the Cohen's D effect size increases, the number of patients is generally a lot smaller. The number of patients we need to find these significant findings are smaller. But we are tending to work with effect sizes of 0.4 and below. So you can see that at around about 0.4, we need around about 100 patients to recruit for sufficient power. But if you remember from the CHR studies, we're down around about 0.1. So if you project that curve higher, that would reach about 1,000. So this is why really we need to focus on pooling our resources because we're just not going to be able to get robust results otherwise. Okay, I'm going to hand over to Marianne.
Change of seats. Hey. Hello, everyone. So now I'm going to go back into what we're doing in Psy-ShareD. So we're working with structural images from different sites. So what that means is that we get a lot of variability in the scanner and imaging protocols. You have different manufacturers, you have different imaging acquisition parameters, you have different field of views, you have a lot of different parameters and effects that are going to change and produce differences in the data. But these differences might lead to biased and inconsistent measurements, which is why we want to do some bit of harmonization in our data to be able to analyze it properly. Now, this type of harmonization can be divided broadly into two. The first one that I'm going to talk about is the image level harmonization, in which basically the idea is that we grab the original structural image in this case. So we only work at this moment with T1 images. So we grab the T1 image, we put it through this harmonization algorithm that I'm going to mention and go a little bit more into detail of the examples later. And then the result is another image. So we get a full image that is already harmonized. Then the other set of general examples of harmonization methods is a feature level. So for this, you need to grab your original image, and then you need to process it to get features. So you get the original mesh, you get some type of measures. It could be either volumetric measures, cortical surface, whatever you want to do. And then you bring it into the harmonization method. And at the end, you get harmonized the same measures that you had before, but harmonized. So I just want to show you one example of a harmonized image type, what we go. So for instance, we're going to talk about one method that is called HACA3. So this image at the left is the original target image that we want to use. And this is an image that we have before harmonization. Now, to be able to compare these two, you can see that for instance, the intensities of the image are quite different. So then we grab this image and we process it through image level harmonization, and then we get something like this. So basically this way you can see that at least the intensities of the image are quite different. There's other things that go into play here, but like visually, you can see now that the intensities are more similar. So then this allows for a more direct comparison analysis in between these two images. So within these two methods of image level and feature level harmonization, we are using 4 different methods. The one that I just explained, used the example before, that is called HACA3, that was published in 2023 by Suetala Group and Hopkins. They use an autoencoder deep learning method, basically where they try to detangle the biological information from the information that comes from the contrast of the image, and with this separate information try to produce a new image that only has information from the contrast more or closer to the original, but keep all the biological information. Now, there's the other image level harmonization method that we use is called Iguane. That's how I'm calling it. I'm pronouncing it, at least. So this method is basically it's a cycle-GAN method for anyone who might know the reference. It's another deep learning method that use a generative network. So within this generative network, they also work in training the data set by using a lot of different images from different sites and images that scanned in the same one. So then within each slices, they can look at the differences, and they can as well separate the biological from the contrast. So that's the benefit of using an image level harmonization, that whenever you're controlling and harmonizing everything, you can be able to separate the contrast from what is biologically actually there. Now, the 2 feature level image methods that we use, we use 2 different combat methods. One that is called neurocombat. Combat is a method that basically works a little bit like a regression, in which they extract the residuals, but they control for other variables, for instance, sex and age. And then neurocombat was one that was created and built on top of the original combat that was made for gene methods. So neurocombat, that they started with gene methods, then they went to DTI, and then they created neurocombat that works basically with cortical measures, and was built specifically for that. And then on top of that, there's this new method that is called neuroharmonized, that is a combat GAN. So it's combat, but it also works with a generative network to try to control a little bit more about and work better in more synchrony with other covariates like sex and age as well. But instead of using it just as a multiplicative or additive way, they use it and they integrate it within this deep learning method. So here's an example where we grabbed 8 different studies from our Psy-ShareD database and repository. And for instance, we extracted the total gray volume. So this is for the original measure. So this is the total gray volume in millimeters squared.
Yeah, millimeters
Cubed. Millimeters cubed, sorry. Had a little. Then you can see how each average measure changes a lot according to the study. Now, all of these studies that I included here are recruited with different scanners and from different places. There are some studies that are from Italy, others from Japan, others from the UK. So you can see that there's a lot of difference, and that difference might be reflecting something that it might be about the group that they scanned, or it might be reflecting differences from, it is reflecting differences from the group that was scanned, but also about the scanner. So then these 2 different methods, you can see how the variability changes, but not as much. So these 2 are fairly new, the HACA3 and Iguane, that are the image-based image level harmonization methods. They do not reduce the variability as much as these 2 do, but then this has to be taken with a little bit of a pinch of salt, because when you use combat methods, you reduce all this variability, which means that you might also be reducing the variability that comes from biological sources instead of just taking care of the scanner. So, but these two methods are a bit more noisy. It's a very complicated procedure that we have to do. So, and these algorithms are just getting validated now. So we're working on it. We're actually working on a paper that is going to hopefully come out in the next few months, where we're doing a comparison between all of these harmonization methods. So look out for that, for the actual recommended one, because I think, as we've seen right now, it's going to depend on what type of analysis you're trying to do. So now that we've covered the harmonization methods, let me show you which type of data processing we do as a group. So we get this imaging data, which is mostly T1 images, and we get the clinical and cognitive data. All the imaging data then gets processed with FreeSurfer Reconol. So this is, as Matthew mentioned before, as Simon mentioned before, hopefully saving you a lot of time, because you can actually access the already processed Reconol data sets. And then on top of the FreeSurfer data set, then we can do the feature level harmonization using one of the two combat methods. And then independently, we're also doing image level harmonization, and then using FreeSurfer on top of those harmonized images. So then what we have available is image level harmonization, feature level harmonization, and sometimes we do have this processing, but that is not for absolutely all of the data sets yet. And then the other thing that we do a little bit more separate is try to do a little bit of a measure harmonization because we get different types of cognitive and clinical measurements that can sometimes be different according to who is getting that data. So now I want to do a little bit of a break and allow people to ask any questions. So I want to make sure that everything is clear so far, because now we're going to move into the more interactive bit. So I am looking at the chat. We also changed the settings, and now you're allowed to unmute yourself and ask a question if you want to. So I'm going to give you like a minute if you want to ask anything at this point.
Can I just ask a question? Well, just sorry, while we're waiting. So in terms of the difference between the image level harmonization and then the feature level, you called it, right?
Yeah.
So do you know why? Because you were saying basically the feature level was kind of reducing that variance a lot more, and you pointed out that there could be a concern there as well. Is there a reason why the image, I suppose, doesn't change it doesn't sort of squash the variability so much?
Yeah, so basically the two image level harmonization methods that we use they use the deep learning methods to try to detangle and separate this biological from the contrast measure. And the way that they do it is that they analyze the different slices. So for instance, you definitely have different biological information in each slice because you're looking at different areas of the brain, but you keep the same amount of contrast. So they use the image information to try to detangle, and then when they rebuild the image, because at the end what they produce is a synthetic image, right?
So you got to get a corrected image out or something, is that?
Yes, exactly. So you get a corrected image outside. So it might keep a little bit more of the biological information there.
Okay, that makes sense.
Yeah.
And just the other question I had was in the measures, you were saying we have kind of the clinical measures. Is that at the moment, is that something where you apply something like combat, or is it more kind of working out? Because I suppose you can do it more from an understanding of what the different measures are looking at, and if there's some comparability between, say, different depression measures and things like that.
Yeah, it's more towards that type of thing. It's not like using like an algorithm.
Okay, so it's more just trying to understand how they relate to different scales and so on.
Exactly.
Okay, cool.
Yes, it's trying to see how they relate to different scales, and basically because sometimes there's different scales that are going to be a little bit biased, so you can do a little bit of a normalization there within each group.
Okay, cool. Okay,
more basic. So for the next bit that we're gonna work on, we're gonna work with something that is called a Colab notebook that is very similar to Jupyter Notebook. If in case anyone knows, can you send the link on the chat, please? So we're going to work with 2 example data sets. These data sets, because we need to keep track and to make sure that everyone gets enough credit to whoever they need to give credit, and we don't have data agreement with you guys. We've modified these data sets a little bit. So we added noise, we included a couple of dummy variables, but most of the data sets should have more or less the trends that they need to have. And then Ruben's going to send on the chat this link, and it's something that you can run in your own browser and in your own computers. So we're going to use the analysis results from 2 free surfer Recon. And we have original data. We have harmonized data from the 2 image level harmonization that I mentioned, and we're going to look into one of the NeuroCombat neuroharmonize, one of the combat method feature level methods, so that you can see a little bit how to do it yourself. And I'm going to move to that. So I'm going to stop sharing this, and I'm going to start sharing the interactive bit. And again, the idea of this is that it is mostly that you guys can participate as much as you want. Let me see if I can mirror the screen here. Give me 1 second. Much better. Okie doke. So we're there, and I'm going to share the screen. Okay, so hopefully, did everyone get the link as well in the chat? Yes, so the link is in the chat already. Hopefully you can see it. So this is a Jupyter Notebook. So let me explain a little bit for the people who are unfamiliar with it. Basically, it's a container where you can have code and text all together. This is all going to be in Python code. Here you have your table of contents, and you can go to each one of these cells and run it by clicking here. This is made by us. It's not by Google, so it's going to give you a little warning. I assure you, we're not trying to contaminate your computers. The first thing you're going to have to do is just run this first bit so that you can run the rest afterwards. This only imports, tries to install the 3 packages that we might need, that we're going to need for the analysis a little bit later. But the rest of the packages are all already integrated here. So basically, here we're going to start importing all the packages from Python that we're going to use, so that we can have access to all of these tools within this data set. Then the next thing that we're going to do is import the data. We have the data set saved here. Remember, this is a bit of a dummy data set. It has real data, but it is also mixed up with non-real data that I'm not going to tell you what it is. We have
just letting
data
you know
from
what
2
I have
different
available. I've got a busy day today.
Sorry, do you have any questions? Okay. Okay, so going back, we have the data sets here. Here we're just importing them into Pandas data frame, which means that they're saved within the Python, and we have access to all of the data there. And then we start having a little look into the data. So here we start printing, for instance, what the groups are that we have in here and the age groups. So if I'm going too fast, let me know. If I'm going too slow, I'm sorry, I'm trying to go so that most people understand. If you feel like it is too slow, feel free to move forward. But when you go into the data set, sorry, I forgot to run this one. I need to run that one to load. I need to run this one first. To import all the packages, you see the little arrow running. Then we import the data set. There you can see that you have the data set well imported, and then we're going to start exploring it. So we can see that we have the group in 2 categories. So here we included 2 data sets that has just schizophrenia patients and healthy controls. The average age of these are 41 and 37. And then we can click on this little thing to describe more or less the ones that are all the categorical variables that we have here. So we can see that we have 46 subjects in total, that we have 2 type of groups. We have 32 schizophrenia patients, and we have 2 biological sexes here in this group, and then these are and there are 26 females. And we do the same type of description in the second data set, which brings us to the summary where we can build this little table showing all of the information that we have, all of the information.
I told
like
you I had 8 minutes. I have a busy schedule. Am I supposed to just put things on hold just to accommodate what you want at the moment? And I'm still really frustrated about this whole thing.
Sorry, are you talking to? Is your intention to talk to the group? I don't think so. Okay. And so, okay. So now sorry. So now the first thing that we're gonna do is checks and do some sanity checks in the data. So the one thing that is seen a lot of the times is the difference between sexes and the brain size. So here we can extract from the free surfer data. We have the sex data, we have the sex information, and we have the segmented volume there. So we can group by sex and describe. So we see that we have females and males, and we can get the mean values with the standard deviation. And then we plot this information, and we can see that we see that it does make sense because females have smaller volumes than males in this first data group. And we actually see that this has a big decoin effect that is 1.3. That is what we usually see in this data set. So it's a good sanity check because these come completely anonymized to us. So it's our way that we check at least that these make sense. And we thought that would be a good example for you guys to see what type of basic analysis we can do. Then on the second data set, we run exactly the same things. And again, we see, but with inverted x-axis, that the females have smaller volumes than the males. And then we see a little bit of a smaller coefficient, but it's still very significant. The next thing that we want to see is to do another sanity check where we have a gray matter volume again versus age. So something that we do know about the brain is that over time it shrinks. the gray matter volume decreases. So we're going to do the sanity check with this second data set where we have X is going to be the age, Y is going to be the total gray volume, and then we're going to fit a linear regression with X and Y. And then we're going to plot this linear regression that we see. And we see how age in the first data group, it decreases over age. And then we look a little bit of statistics, and we see that this is somewhat significant, but this is more or less the type of things that we see because obviously everyone changes. Everyone has different brain sizes. Then we see then we do exactly the same analysis for the second data set, and we see something similar. Data set 2 has a lot more data points, and we see a more clear trend here, or we can see that it's a lot more significant in this case. Now I want to see if there's any questions at this stage. Give a little pause or a little bit of time. because in the next, we're probably not going to finish looking at every single bit of it, but at least you're going to have access to the notebook. So you can use these tools. Hopefully they can be helpful as an example of something that we can do within the Psy-ShareD, and we're using the Psy-ShareD data. The next thing that we're going to see is try to do a little bit of a comparison, comparing information that we get, comparing data that we get originally from the data that we get from the neuroharmonized, that is the combat GAM. So we use those a little bit interchangeably, apologies. The HACA3 and Iwana imaging level harmonization methods. So using this, we're going to do a little bit of a SVM model to classify and differentiate between healthy controls and schizophrenia patients. And then we're going to see whether the harmonization is aiding in this type of classification. Now, I want you to keep in mind that these are just two small data sets, so we're not going to get the most fantastic type of the most fantastic classification, but at least it's going to be a little bit of an introduction, so you can see what type of effects we're looking into. So the first thing that we need to do here is do a little bit of data preparation. So we want to, our data sets come with sex as M and F. So we're going to change that into numerical methods for the type of functions that we're going to use. And we're going to change the target from the group from the healthy controls and schizophrenia into zeros and ones, so we can do this binary classification. And then what we're going to do is we're going to load and combine both data sets so that we can run them together and merge them. We're going to get rid of a few variables that we don't want. For instance, we don't want the group information within the data that within the the data that we use to train the model, because it's a categorical value. We're removing all of these categorical values, and this one that is a bit useless because it's just added when we loaded the data set. And then we create our X that is going to contain all of the features that we have for each data set, and our Y that is just the target, which is just the diagnosis of the groups. Now, and I'm going to run that, and there we go. And now, which models we're going to use a few tools here that it's important to clarify. So we're going to use a cross-validation. Apologies if you're not very familiar with this, but it's basically a method in which you grab the entire data set and you divide it into subgroups, and you train with one, you remove one subgroup. So say that you divide it in 10 equal parts. You remove one of those 10 equal parts, and with this other 9 parts, you train the model and you test it with this 10th part. And then you put this one back, remove the second part, train it with the other 9, and test it with this one. And you go like this, and this is a way that you can use basically all the data and see how generalizable it is, because if by chance I get an outlier when I extract it here, then this prediction is not going to be good, so we're going to just test it out. Then the feature selection is because we have right now in these columns and this data that we have over 200 columns and features that we have. And then we're going to use a tool called select k-best based on mutual information. So basically we calculate the mutual information or the amount of variance that each one of these columns have. And then we say, just select the k amount that has the most variability, and therefore is more likely to be the most informative for our classification. And then finally, we use certain evaluation metrics: accuracy, precision, recall, and area under the curve. I'm going to focus when we're talking about a little bit more in accuracy, just because it's basically how accurately you can predict that someone has a psychosis schizophrenia diagnosis. But we also have the other 3 because to show you that there's not particularly a big bias. So the first thing that we're going to do is that we're going to define the model for the cross-validation. Now, normally you would do a cross-validation with 10 or more. You can even do as many subjects as you have, and that's called a leave one out cross-validation. But in this case, because we're running it live, I'm going to reduce it just to 2. The results are going to make a bit less sense, but we do have the results for when we run it with 10, but this way it will run a bit faster. So I'm going to run this cross-validation here, and then I'm going to run the classifier. So we have the SVM classifier that is just using this SVC function with a linear kernel and this random state, just so we get the same results every time that we run it. And then we're going to do this K stratification k-fold that is basically creating the groups, separating the groups as I mentioned before. And then we have these variables that are just going to store the accuracy, precision, recall, and area under the curve. So then we perform the cross-validation by iterating them k number of times. In this case, it's going to be 2. In your homes, you can run it with 10. The beauty of this type of notebook is that you can run and modify the text, and it's not going to affect what I'm doing here. It's only going to be in your computer. And then we're going to split it into train and test sets, and then we're going to select our features for those training sets. Then we're going to select those here, and then we're going to just fit the model, because this is just saving the selected features so that we can look at them afterwards. And then we fit the model, and then we try to predict the results using the testing data. So I'm going to run this, that is just the function, so that we can run it afterwards. And then obviously we save the results. So first we're going to run this using the original data set. So the data that we had looked at before, and then we have the combined data set. So then we run the classification here. Keep in mind, this is only k-fold 2k to cross-validation groups. So it might take a little bit. There we go. And we see that the accuracy, at least for the original, is 64%, which means that it accurately predicts that someone has schizophrenia 64% of the times. Now we're going to run the same for HACA3. I'm just going to run it quickly so that we don't have to wait as much that time. So for HACA3, we have exactly the same data set, but the values that we have here have already been harmonized in the image level and then processed through FreeSurfer before we import it. We have more or less the same data size, and we wait for the classification here. Here. And we see that at least for the HACA3, we have a very similar accuracy of 62.5%. And then we're going to run it for Iwane. We similarly with the HACA3, these have been images that we have pre-processed and harmonized through the Iwane algorithm, and then processed through FreeSurfer. And now we're going to run the classification here. Just going to change that. And then we see that the Iwane classification actually does better than the previous 2 ones, in which we have at 69% classification accuracy. which is a lot better. And then we have, then we're gonna run the combat harmonization. So for this, we need to load the original data. So we load the original data that we have here, and we add a variable of site because we need to tell combat that these are two different, these data were scanned in two different sites. Okay, so then we combine the data sets. We have a different variable that is the covariate variable. And we have a data frame that we want for combat, which basically has all of the information, all of the features, except the covariates that we were looking at before. And then NeuroHarmonize, that is the one that uses Combat GAN, uses harmonization learn. So where you give the X data set where we have basically all of our subjects and all of the features, and these covariates that contain only the information of the site, age, and sex. You can add other covariates if you want as well. And then it releases, it gives you back the data, the model and the data that has already been harmonized. So it's super easy, super fast to run a harmonization. And now then we put it back together into our data frame. So then we can do our analysis. So we just run it there. It's very quick. And then we run our classification with the Combat GAN NeuroHarmonized tool. While it's running, please remember that you can put your questions in the chat. Ruba next to me is looking at the chat and ready to answer any questions. If you have any afterwards, I'm happy to also answer by email. And we can see that the Combat GAN also has a accuracy of 67 almost %, which is better than the original, but not as well as the Iguana that we had before. Now, the results that we had displayed before are only when you split the data set into two groups. But we had already run it beforehand using 10 cross-validation, 10 subgroups, and we got these other different results where Iguana was also a little bit better, also performed. And we have a better AOC, area under the curve score for Iguana in this case, because you have to look at all of the measures, even though now I was focusing just on accuracy, just for the purposes of the demonstration. And you also can run some analyses looking at how the area under the curve changes depending on the fold number. And you can see how it goes sometimes improving. It can start very bad for some of them, but then go improving, hopefully, with the number of folds. And this improves a lot more when you include even more data, which is why we want to make this data set available. And I give you this little code for the AOC scores that it's going to make more sense when you run it with more folds, because here it's going to just produce 2 data points. And then another very interesting thing that you can look at is the top 10 features that you get from the feature selection. And you see in the top 10 features here for the original data set, then for the HACA3, then for the Iguana, and then for the Combat GAN, where you can see that we sneakily added one data point, one feature to the columns that is not just feature image level, but it's also from the cognitive information that we have, the linked cognitive information, where we have the WASI matrix task and the results from that one. And we see that this little measure is available it was selected for the original one, for the Iguana, for the HACA3. Here's the original one, HACA3, Iguana, and the combat. Not in the combat, but at least in these 3, it was selected as one of the features that was going to be most informative. Then you can go and look as well into the importance and the weights of the coefficients for the SVM. That is also going to be even more informative into how each feature contributes to the data. And you have the code there for running this same type of plots. I'm not going to run it at this point because it's not going to make much sense, because we only have two k-folds. So in conclusion, what we wanted to do here is just demonstrate the type of analysis. Like, this is like the very basic that you can start doing, the very basic type of comparisons that you can start doing. And we wanted to provide some tools and show a little bit how you can use, from your own computer, you can just like use the features that we have already selected for you and run your own analysis. So it was basically to give you a little bit of a introduction and showing what type of analysis you can do.